Thanks to the audience for participating to this webinar. We will talk about the NETRI technology and the applications that you can use for. There will be a presentation first about the technology of the microfluidics that we have developed and that we are proposing. Then we will focus on two aspects of the presentation in the 2D architecture and then in the 3D architectures. That would be more or less the 20 minutes and 20 minutes for each part. Then it will remain time for question and answers. That will be the last 20 minutes of the hour of the webinar. I will be also for the question and answers, I will be assisted by my colleague, who is the field application scientist, Dr. Nena Salman, that you can see maybe on the screen. And then she will help to answer some of the questions that KG will address in English to us. So let me start first by a few words about the company, about NETRI. So NETRI is a French company that has been founded in 2018, and we are based in France, in Lyon. That's a town you may know, not far away from Geneva. So we are about 30 plus people, and we are working in the field of, I would say, broadly, neurons and neurology associated with some applications. We work in an environment which is a laboratory where we have two laboratories, one laboratory for the production here of the devices, and then we have other laboratories where we perform services, where we do drug testing for customers worldwide in our biology labs. So for your information, we're moving into a much higher building in a month from now, and we will have 10 times more capacities also in terms of production and services. So we're going to focus now a bit on the architecture where we started with, which is the 2D architecture, which is what you see between the two fingers of the engineer. This technology has took us 4 years to develop and to be industrialized. That means that at the end of 2022, 2 years ago, we have started the industrialization of this architecture, meaning that 2 years ago and up to now, we have the same architectures and we have the same process of manufacturing because we target high throughput devices and high throughput production also. So, and that's what we are. So we are define ourselves as an industrial startup. So this is here the unit of production, as you can see. So it is composed of 4 identical chips, 3 and 4. And then if we look now at one chip, this chip here is composed of 6 wells and composed of 3 channels. One channel here, a second channel in the middle, and a third channel here. So each channel has 2 wells, an inlet and an outlet, knowing that there is no directionality into the channel. So you can decide this is the inlet or you can decide this is the inlet, doesn't matter. So basically our chip is composed, as I said, about 3 channels. So you can do co-culture or tree culture in our devices. And what is important is that we are capable to co-culture or tree culture the cell types separately. There is no mixing of fluids between the channels. So the channel 1 has a specific cell type with a specific media associated with that will never mix with the second channel or never mix with the third channel, which will have another type of cells. The reason for that is the middle channel here acts as a critical wall. We have devices and patents for that and know-how. And so basically, with a combination of overpressure and microfluidic design, we are capable to guarantee that there is no fluidic separation, mix between the three channels. So that's very important. And the reason why it is important is because in all anatomy, the body has different organs and also different membranes, and all is together but separated. For example, if you take a sensory neuron here, that's the sensory neuron which go to the spinal cord here, and then it is separated, for example, from the dermis, whatever, the epidermis, etc., and dermis. So from your skin standpoint, you will have a sensation that will be seen from the skin upper level, the epidermis, and then you will get the transmission of the signal through another vehicle, which is the sensory neuron in that case, and then will go to the central nervous system and give some information and back into the point where there was, for example, some inflammation, irritation, or anything as a parameter. So compared to the usage of what is usually done, which is a one-well situation where you have a mix of different cells, what we will do with our devices is we will separate those different cell types, making them work together but grow separately. And this is what I'm going to talk to you a little more about. So if you remember back, then we have three channels, one here and the second and the third. They are all separated, okay? So what we will do, we will enable the neurons, for example, that are seated in the channel 3, as you can see here, to grow and to project into the channel 2. So how do we do that? I will show you just one picture by imaging just afterwards. Look at this. Here we have seated the sensory neuron on the left. Then we have a projection of the cells of the axons in all directions from the body. And then at the interchannel, which means between the channel 1 and the channel 2, so at the interchannel here, we have designed micro channels, so very small section of channel, about 3 by 5 micron section, and a length of 150 microns or 450 microns, depending on the architecture. And what will happen is that the axons, which are the only one to take these micro channels, because the fluids cannot go inside, the micro channels will be used by the axon to project as a guide, and they will basically cross using those micro channels from this channel 1 into the channel 2. Okay, so like a water, like a car that goes under the water using a tunnel. That's the axon projection that they will do that. So there will be no mixing of fluids, and they will cross and arrive to the second channel. In the second channel, they are free to operate again, like in the first channel, like in the third channel. They are free to operate within each channel, so they can grow in all directions. However, there are some of them that will also continue their journey and arrive at the interchannel between the channel 2 and the channel 3. One more time, as we have microchannels, same size, same dimensions, etc., then the axons will continue its projection and arrive in the third channel. So if I summarize, you will have endings here of sensory neurons, while you have the cellular bodies on the other side of the channel. So you can help axons, you can help neurons to project their axons and finish their endings on the other side. And that's very powerful, because that's where you're going to start, for example, to test drugs on the endings, which is generally what we feel. Or you can test for certain medicines where they work more effectively on somas. You can adapt and do drug testing on the bodies itself, on the somas. So let's come back now to this slide. So this is a summary of the slide here. So here again, the sensory neurons are on the channel 3 in the image. They were on channel 1, and probably need to modify that for coherence. So what it means is that if you address some other type of cell, a type of cell in the channel 1 here, like keratinocytes, and if you put sensory neurons here on the channel 3, then you will have the axon projection and innervate the keratinocytes. So you can claim that you have at least a proof of concept for innervated skin. Okay? And that's basically a micro representation of some functionalities of organs that are, representing what we have also, natural compartmentalization of our body. If this is understood, then this is, because I insist a bit more on this slide, this is very important for you to understand the rest of the presentation. So I hope this is clear, my explanation on that part. And if you have questions later on at the end of the presentation, feel free to come back on that. So as such, this chip with 6 wells and 3 channels is represented here and 16 times in this configuration. Okay, 8 here and 8 chips over there. 1 chip, 1 condition, 1 experimental condition. If you want to replicate, if you want to have a replicate of 4 or 8 or 6, then you will need to use 6 chips to replicate the same conditions. The beauty of our technology is that it's a very low entry point in terms of easiness to use, and also from an equipment standpoint, it's also very minimal. I will explain that why. The first reason for us to have those benefits is that we use a material which is PDMS, as you see, it's quite transparent. The PDMS is made for, it's like a silicone type, and it has, at the condition you manufacture, you control well the manufacturing process, which we do, then you have 2 main benefits from the PDMS. The number one, is its transparency, its ability to conduct light, so to characterize by microscopy or by imaging, generally speaking, any devices. And you will see in the presentation that we have a lot of examples talking about that. The second advantage of the PDMS, which is a great advantage, it is permeable to gas. That means that the oxygen goes in and out, which means that the cells that are seeded into our devices can breathe without pumps. So the device that you see here on the picture is the device ready to use. You just need an incubator. So you have protocols that we have developed where you can, condition the surface before, prepare the surface of the PDMS before to seed the cells, and then you will basically nurture and feed the cells every second day, which is very convenient, and up to the culture maturity, which depends on the cell type. It can be for iPSC and let's say large cell type example from 14 days to 22 days. Okay. The last thing that you can enjoy with our devices is the ability to maintain the moisture level into the during the whole cell process. We have here labyrinth where you can add water, and this water will help to keep constant the degree of humidity into your device, knowing that we have a top lid here that will also close this part here. So I hope this is clear. So let me now come to the next topic. So you can, as I said, you can do imaging, you can do a lot of readouts, and I will summarize that in the next slide. There is another capability in our device, is the ability to measure the electrical activity of the neurons in life non-destructively. The beauty of our device is that we can add, it's an option, it's not mandatory, But for the same architecture, you can have without and with multi-electrode arrays, which we call a lot of electrodes for each chip that will be able to measure the electrical activity of the neurons. So this chip here is basically bonded into our facilities, into our chips. And then now if we focus on one chip here, that's basically what you will see. You will see a lot of wires and electrodes, around the region of interest, which is where you have the most concentration of cells due to the thanks to the protocol that we have developed. And then if you see a bit bigger here, you see the nature of the mapping of the electrodes. So you can have up to 48 electrodes depending on the architectures. You can have up to 48 electrodes mapping the region of interest. Additionally to that, you can see there are 3 electrodes here on the left out of the channel 1 and 3 out of the channel 3. These 3 electrodes are reference electrodes that make sure that we see, there is no leakage, for example, and that's basically a very important thing. And you can see also that we are having microelectrodes in the interchannel, so not in the channels, between the channels. And you remember, this is where you have the microchannels, those 3 by 5 micron section microchannels where the axons project inside. And this gives us extraordinarily good information because we have a very high amplitude signal versus the noise ratio. So that's very interesting for us. So this technology is compatible only with an equipment that is present in the market from the company Axion Biosystem. And some of you have used maybe or are using the MEA, and they may know the world leader Axion Biosystem and their model, which is called Maestro, like master, I would say in English. And that's basically the compatibility of our device on this machine. There is no other compatibility as we speak of all technology outside this equipment. And it's a plug-in, so plug and play, so that's very easy. So the idea to measure the electrical activity of neurons is to create a database, a digital library, where we will collect and organize the data and find a way to discriminate certain parameters versus others. And so to be able, when you compare a reference drug, which is, let's say, in blue here, and then if you address, for example, a drug testing here on the neurons, or on keratinocyte with innervated, then you will get a response with several parameters for each response. And then you will test the yellow compound, which is the compound to evaluate. And then you will be able to compare this to the other references. So the really good point about the technology is that we have comparative data using the electrophysiology. And that's generally what our customers are asking. They say, I have this reference internally that works, or that the literature talks about, and then that's the one we want to have as a baseline, as a data, electrical data to show, and then we will introduce a new compound we want to evaluate. So we are actually now internally capable to do it in a let's say in a first version of digital library. We are going to expose this and present it as a service to the market and to the community in less than a month from now. So it is a major move forward because we are a bit in advance compared to what we schedule. So before the end of October, we will have we have data and we will be able to present them and show the power of it. So as a summary of the readouts you can obtain with our devices, I mentioned to you the great transparency of the PDMS, which allows us to make any type of imaging, whatever, if it's immunofluorescence, calcium imaging, live staining, live data sets, and so on. We have also the possibility, and most of the customers are doing that, to address any type of characterization using the supernatant by collecting them. So we collect supernatant from the wells, and then we can, after that, have ELISA, analysis, Western blot, that can be done not in our lab, but outside for, let's say, protein dosage, for example, and so on. Additionally to that, we have the electrophysiology, which as mentioned. So this is very powerful, and I will discuss a bit more about some other readouts which are possible in the 3D architectures. But for the moment, for the 2D architectures, you have a great scope of readouts possible, which basically cover the entire need from our customers so far. So that's the technology. So now what I would like to do is show you some examples of studies that we have done without electrophysiology, so with image characterization, and also with electrophysiology, so with, as it said, electrical characterization of the neuronal activity. So let's take one example here. So we are in the 2D architectures. Then you can see that we have grown some motor and sensory neurons into motor neurons, into our device on the left, and then we let them grow, okay? This is a very lengthy micro channels here that helps to have synaptic isolation capabilities. So when the network, I would say, of neurons is in place, then we will address axotomy, chemically speaking, to cut in the middle channel voluntarily the neurons extensions, okay? And then we have done this with motor neurons. We have done this with sensory neurons also, introducing sensory neurons, extension of the sensory neurons. So when all is in place after 22 days of maturity, then we apply a chemical axotomy. And then we will look at the results of that. So, for example, we have here used, the different molecules, the staurosporine and also the nocodazole, to evaluate the regrowth and also the degeneration of here the sensory neurons. What does it say? It says that, for example, we are capable to characterize the mean length of the recovery from the different concentrations that we can apply, for example, for nocodazole from 1 micromolar or 0.1 micromolar, and also the vehicle that is used, and also the staurosporine, which is here for recovery. So you can characterize by imaging the mean length. You can also characterize the number of axons by imaging. You can characterize their maximum length. You can characterize their sum of lengths, etc., etc. So you can semi-quantify the effect of regrowth from our devices using, for example, the different molecules and concentrations that we have here. So what we show here in this data is that there is an inhibition of neurite outgrowth of nocodazole at 1 micromolar. Okay, and at also 10 times lower concentration. So this reduces the number of axons and the total sum of lengths without affecting cell viability. So you can conclude certain things from motor degeneration diseases where you want to address certain drugs for that effect. Okay. Now you can also use the electrophysiology to assess, for example, the axonal activity. Okay, so if you consider here the different electrode type here at different position, then you would see that you can elaborate the electrical impact of the regrowth of the axons. You can also confirm functional recovery. So you can see that, for example, when the axons have regrown, they are capable to function back again as they should. And so that's a very functional information electrically wise that shows not only that the imaging is giving you the information, but also the capacity of the electron to behave electrically as it should be, or if there is an electrical impact. So it's complementary, but it's also very powerful. So some customers address those questions directly by asking, response from the MEA features and MEA capabilities, and some others will start first with imaging. That's really depending on the customer's request. Another type of application in the oncology field is, for example, the pain after chemotherapy, what we call chemotherapy-induced pain neuropathy. We have, when we unfortunately have to treat tumoral cells and using, chemotherapy, and most of those molecules are known, like the oxaliplatin or paclitaxel. Those molecules have a first aim is to reduce or destroy the tumoral cells. Unfortunately, there are some drawbacks and counter effects which give the sensory neurons higher sensibility, and so you will feel more the pain even if there is no real pain induced. So this is a very dramatic situation for the human beings treated, and so there are a lot of focus to find what are the best drugs, best concentration of drugs that would minimize or eradicate this hypersensibility of the sensory neurons. So what we are going here to present, I'm going to present you sensory neurons again in the channel 1 with their extensions on channel 3. And basically, depending on the molecules and their ability to act on the neuronal sensory network in our body. For example, the oxaliplatin is known to be effective into the somas, while the paclitaxel is known to be effective on the projection of the sensory neurons, so on the extensions. So what we will see is some examples here of paclitaxel different concentrations. So we're talking only about imaging here and beta-3 tubulin marking. And then what we have is we're going to look at two parameters. For example, the surface coverage, is the surface coverage of our endings of sensory neurons, are they evaluating? And the second thing is also the debris, what we call blebs. So basically the degradation of the termination of sensory neurons gives some small nodules along the axons that show that the sensory neuron is suffering. And basically what we show here is, depending on the concentration, there are some effect, like for example here, the 5 nanomolars is giving a small effect, and so which will show that there should be no major drawback using this versus higher concentration. And for oxaliplatin, we have also conclusion to take from the variation of concentration here. Okay. So now, if we take the same experiment but using MEA technology, what we will have here is we will have the capability, as I said, to measure the electrical activity of the neuron. So, for example, with variation of temperature, then we can see that the sensory neurons treated with oxaliplatin seems to react, more to low temperature change when compared to vehicles. That's, for example, one kind of information you can obtain here. On the paclitaxel side, we can see that it increased the electrical activities of the sensory neurons, and then there is basically a detection of acute effects without inducing cellular degeneration, for example. So that's the type of information you can get with a powerful MEA. So as a summary, you can see here that there are, several possibilities. I talked to you about PNS, and we mentioned the sensory neurons, but we have also data and experiments using CNS, like gluta and GABA, in certain CNS disease, like the neurological disorder disease. So I'm going to be a bit faster here. Just maybe one comment that we have the capability as a service in our device, in our laboratory, to measure the viability of the cells before to do the drug testing. So after the cell maturity, we're capable to measure the cell activity before testing, and it's non-destructive. So that gives us extraordinary confidence into the viability of the cells and our devices before the testing. Another thing that we are capable to do with electrical physiology is because it's non-destructive, we have the ability to, when we test in fast response, we test the vehicle first, see there is no interaction, then we test the compound on the chip, and then the compound X to evaluate, and then we will have a positive control testing afterwards to make sure that the cells that have been tested on the compounds have the right behavior, and that's give us another indication is that if after the positive control the baseline of activity is coming back to where it was before, that means that we can add another experiment, another set of drug testing into the same chip, so we can have a chronic response based on this technology. So that's very powerful. So now we have discussed about the 3D. I have a few minutes more to discuss about to talk about the 3D. Sorry, now multifluidics, and the new product that we have launched this summer is Duplex, and then we will talk a bit more about that. So the architecture of the Duplex is based on the central well, where it's an open well, so you have a certain size. It's about 5 by 6 millimeter and a height of 4.5 millimeter, so quite a large dimension, where you can have you can put any biological material. I would say it could be 3D. It could be an organoid, it could be a spheroid, smaller. It could be explant, etc. So, and what we have is we have designed, so the two wells here, what we call side wells, are linked together with a perfusion channel here beneath that are linked together. So if you follow my pointer, this is the same channel. What is the beauty of it is the channel below is not fluidically in contact with the bottom of the well, because there is a membrane here, a porous membrane of 0.4 micron size, pore size, that will isolate fluidically the perfusion channel from the central well. That's what you see here, the nanoporous membrane. So we will enable interaction from a molecule in ionic standpoint, but not from a fluidic standpoint. So we want, again, to replicate membrane situations and environment into our body using our technology. So another way to look at it, irrigation channel below, a central well, and a porous membrane where the biological material will stay on in the central well. So that's just the configuration. So again, that's 96 well plate format that you have basically 96 well plates. So that means this is fully compatible with lab equipment, like the 2D architecture. I may not have mentioned that earlier on, but that's also a great advantage. So it's fully compatible with robot handling, liquid handling, and we use it in our laboratory as a service to automate and to, for example, do PK profile and drug dosage in oncology studies and benchmarking chemotherapy versus the tumoral cells progression. So this is the architecture here. Then you have a bit more information about the volumes, about the volumes of the each well, the channel below, and the central well. Okay. So for example, the well is about 140. The lateral wells are 140 microliters. Okay. And the channel below is about the same volume, so about 145, okay? Okay, so now I want to give you a few examples of applications that we have conducted. The first one is talking about brain organoid on chip. So we know that they are in the market, already extensive studies on in vivo and on 2D human cells, even 3D human cells. So we know also that there is low predictivity using the in vivo or 2D human cells for brain organoid. So for brain on chip. So the 3D architecture, I would say, is more suited for this type of answers to questions to answer. Sorry. So basically, what we have decided is we have conducted those two studies because we think that the microfluidic devices will bring a more controlled flow of the fluids that you are using. For example, the media that you are using for nurturing and feeding the organoids. We have also the idea is to also transpose our capability of readout and automation processing into this field, and I will show you some examples of that. So we have conducted, over the last year, 2 different studies, and the second study, as well as the first one, I've conducted to some posters that are available on our website and that we have presented, and that we will present also in SFN in Chicago in 2 weeks from now. So this is basically the idea is that we have put the organoid, brain organoid, into our central well. Then we have put here, some media, and then we have used the perfusion compartment, which is this compartment, to feed the central well where the organoid is. Okay? So this has been done statically. There has been no rocker, no need for moving the devices during the experiment. And what we have seen is we have seen on our duplex well, compared to the typical 24 well plate or negative P96 well plate, that we have seen that on a P24 plate that we, when we grow organoid, let's say, it is what we expect. When we use 96 well plate, then we see that the organoid is far too small, which is not corresponding to what we need. And then what we've done is, case 1, we have feed from the bottom and from the top always. So every time we were changing the media, in the central well and on the lateral wells. That's number 1. Number 2, we were using changing the media from the lateral wells every second day, and once a week we were changing the media in the central well. And then the third one is the one where we change the media only from the side wells, okay? And then you can see with different time point introduction of the EBs that basically the best results we have is on number 3. Does not mean that number 1 is not good. It's good also. 2 is good also. It's even better. But 3 gives, you can see more coherent shapes on that. And now if we look at the size of the organoids, we have done that on 21 organoids. What we've seen is that in green, this is into our devices, the duplex well, and compared to the P24 well plate in blue. So each point here for a time being is basically 21 organoids, so 21 results. And what we've seen that with much narrower results, so which give us much more repeatability and a very predictable organoid area, so organoid size over time. And then when we look at the cytoarchitectures, then you can see again, if you compare the 24 well plate, 96 well plate, and the 3 different conditions of medium renewal, that number 3 is just giving excellent results in terms of size, but also capability to from the rosette to express for the organoid to express neurological standpoint. So that's very, very interesting. So we had very good traction on that one. So that's the summary of this slide, and this slide summarized the conducting of the study here. So it gave us good perspective into projecting it into blood-brain barrier. It is not what we have done yet. This is just a first step. Then we have to work into and with customers also astrocyte, pericyte, and endothelial cells to mimicate the membrane, and then later on add the organoids, which is something that we are open to discuss with customers. Last but not least, we have also the capability to measure the permeability of the membrane using tear measurement. With all devices, we have developed a specific adapter, very simple, that goes on top of our devices, which allows an electrode that we move manually from one chip to another chip. And we have, let's say within 1 minute, we are capable to address 16 results of tear, very repeatable. And if you look here at the images, you have 6 results, 6 times, and then you can see the high repeatability here. So that's a great advantage also using tear compatibility with a very low cost standpoint, and knowing that we work with a world leader world precision instrument for their metrology systems here. Okay, last, very quickly, it's the pre-vascularized skin proof of concept. So basically, what we have done here, we have started to do first experiments on how we could mimicate innervated, vascularized, skin. So we have introduced keratinocytes. So it's 2D culture into a 3D well, okay? And then we have put endothelial cells from the side wells here in blue. And then we have basically looked at the effect of having endothelial cells and the differentiation that you can have with endothelial cells first, and then also the morphology of the keratinocytes that is changed using the endothelial cells. So what it does say is that there is a passage of molecules from one compartment to the other, so through the membrane. So that's very important, because again, that's the proof of that the device can mimicate a skin innervation and skin vascularization here. So last point is that we are going to launch soon an additional channel here that will help, with micro channels also here, that will help to have the axon projecting from this type of a channel into the central well. So we will have innervation, innervation. So you can see innervation. And then we will have, vascularizations, and then innervation from the lateral one. Finally, we have also other capabilities, but that will be, let's say, next year, where we will be able to connect together the chips and use, for example, immuno flow or endothelial flow between different chips and having this common using perfusion systems. So as a summary, we have the technology because we develop everything from A to Z, and we have the capability in our laboratories to screen or co-develop models for our customers in our NETRI labs. So thank you for attending and listening to the presentation. It has been a bit longer maybe than expected, but I'm ready to answer any questions that you may have.